Hi, I'm Stephen Beebe, and I'm a professor at the Frank Reedy Research Center for Bioelectrics in Old Dominion University in Norfolk, Virginia. And I've been working on this project with nanosecond pulse electric fields for almost 20 years now. And I began because I was interested at the time, before I even knew about this, in cell signaling, and that basically um, how cells change their behavior in response to stimuli from the outside. For example, a hormone or a drug that affects a receptor that changes the behavior of a cell. And I realized that pulse electric fields was a unique stimulus that can do the same thing. And I was, at the time, one of the signaling things that I was studying was apoptosis. That is, how cells die. And we realized that around that time, or actually before then, that cells died in at least two ways. It's a little more complicated, but simply cells can die by necrosis, where they just basically fall apart, burst, release their contents and cause inflammation. But cells also die by a process that's more organized called apoptosis, and thousands or millions of our cells are dying every day by this process, and we don't have inflammation because of it. So it's an organized way for cells to die. Analogy might be a building that's being demolished, and it's exploded and it collapses. That would be like necrosis. The other analogy would be, instead of that building exploding, you start taking apart piece by piece and saving parts of it that you might want to use someplace else. And apoptosis really does that in a sense. It takes the cell apart very in an organized way. One of the things that happens is a stimulus from the outside tells the cells that it's time to die, or something happens that the cell can't resolve or can't overcome. So there's enzymes turned on inside of the cell. They're called caspases, and they basically start taking the cell apart, piece by piece, breaking down proteins and ultimately breaking down DNA. They also cause a signal to be sent to the outside of the cell. It's called phosphatidylserine. And we have a tool that will bind to phosphatidylserine called annexin. And that phosphatidylserine identifies the cell as one that's dying and it's ready to be phagocytized before it breaks and causes inflammation. So what we found with the pulse electric fields is they could in fact do that. In some instances they can cause necrosis in some cells, but in many cells they cause the process of apoptosis. And this is important because this is one of the things that cancers avoid. They avoid caspase activation. They can prevent that and prevent themselves from dying. So in this instance, the pulse electric fields induce apoptosis, and the cells die in an organized way. And this is something that's been very important in terms of this technology for cancer treatment. So I'm really excited and still excited about working on this project. So I've enjoyed it very much. It's a great way to live.